All right, good morning. Thank you so much for coming out so early. I hope you had some great breakfast. We have lots of it in the back, so if you haven't, feel free to grab some more. We're just gonna kick this off, and I wanna welcome you all to our annual breakfast seminar, this time on ASMS 2025, and we are very happy to have an exciting program for you today. We're going to be introducing the latest version of CHIMERYS 5. Martin is going to do that. I'm going to be talking about the latest updates to the MSAID platform, and then we're going to be stopping with the product presentations and going to the scientific presentations. I'm very happy to have Dr. Sarah Sipe here from the Parallel Squared Technology Institute, who's going to talk about how she's using quantitative single-cell proteomics aided by parsed acquisition. And we have Edgar Martinez Sandoval, who's a PhD candidate here in Johns Hopkins University in Baltimore, who's going to talk about how he uses our platforms to do limited proteolysis. And before we dive into that, just a couple of announcements as to our scientific program. We were very lucky this year to score 3 oral presentations, 2 of which are today. So Lizzie Mamisashvili, our machine learning engineer, will present updates to our inference model today, this morning in the informatics innovation session. Martin Freno is going to be talking about FDR control in DIA in the DIA session this afternoon. And I'll be talking about applications of our scalable MSAID platform on Thursday in the peptide ID and quantification session. In addition to that, we have a lot of stuff on our plate today. We have 2 posters today. We'll be talking about the spectrum-centric analysis of timsTOF data with CHIMERYS, and we have a general overview poster of the advancements in CHIMERYS, our universal search algorithm for proteomics. And with that, and without any further ado, I would like to introduce to you Martin Freno, our Chief Executive Officer, who's going to be introducing CHIMERYS 5, the work of a team of a multitude of developers over the last half a year. And the stage is yours.
Thanks, Daniel. All right. Also, warm welcome from my end. Thank you very much for enjoying the breakfast. We are surprised by what is offered here. So feel free and maybe, pack something for later. Lunch might not be as good. But yeah, just to start us off, I'm going to tell you a little bit about what is new this year with our universal search algorithm, CHIMERYS. And to get us started, CHIMERYS is not the only software that we develop. We develop 3 different pieces of software plus the platform. And these are inference rescoring, and CHIMERYS. Inference is our deep learning framework for peptide property prediction. Inference rescoring, as integrated in Proteome Discoverer, is used to rescore sequence results. And CHIMERYS is our search engine, which is also the star of the current presentation. Just to give you a bit of a background on where we started and where we are going with CHIMERYS, just a little bit of sort of a history lesson, so to speak. CHIMERYS was released in version 2 in 2023. The initial version was released a year prior. Back then, we already had the capability to analyze DDA, DIA, and PRM data, as well as phosphorylation as our PTM. There was also oxidation of methionine, but phosphorylation as a biologically relevant modification in this sense. And we were relying on PTM localization via PTMRS, a node in Proteome Discoverer. In CHIMERYS 4, released last year, which is integrated in Proteome Discoverer 3.2, we spent quite some time in making the processing faster. So faster processing, we added PTMs, acetylation and ubiquitination, and native PTM localization. So basically localization based on fragment ion intensity predictions. And this year, essentially, it's a major leap for CHIMERYS. On the first one that I would like to mention is that We introduced a so-called new discovery mode. What that is, I will go into a little bit of detail later, but that boosts identifications at the peptide level by up to 30%. It increases data completeness, it increases quantitative accuracy and precision. We added 20 new PTMs to the host of PTMs that we are supporting, and many of them will be very interesting to you. If you want to know what these are in detail, keep an eye on the slides. I have a very tiny slide at the end where you see all of them. We also introduced HLA class 1 support and, as Daniel mentioned, DDA and DIA passive support now as well. And I want to start with the last point because that's a first for us. As you know, that we've been sort of exclusively processing thermal data for quite some time, but now it is also possible to process timsTOF data, and here we have a comparison on a 7-minute DDA run, DDA passive run of CHIMERYS versus 3 other search engines on that same data set. And as you can see, CHIMERYS identifies more precursors here in this case than MS-Fragger, but what is very encouraging is that the overlap between the 4 search engines is very big, and the second highest bar chart in this upset plot is the overlap between SAGE, Fragger, and CHIMERYS. So it is safe to say that DDI passive is now a possibility with CHIMERYS, so now you can actually make use of that deconvolution capability in timsTOF. If you want to know how we deal with that data in detail, feel free to stop by the poster and you get more information on how we can actually spectrum-centrically process timsTOF data. On the DIA front, we have here a comparison on a 7-minute DIA passive run of Diane versus CHIMERYS. Again, very encouragingly, the overlap is the biggest bar here. We identify slightly fewer identifications than Diane, but what is interesting is the huge non-overlap. So there's a huge opportunity still to identify more precursors when you use both search engines than if you were to only use one. That is to say for timsTOF. I want to switch gears now and tell you a little bit about, or give you a little teaser on Lizzie's talk later, and that is how can you actually teach a machine learning model to understand chemical structures. Because fundamentally, deep learning models are limited in terms of their capabilities when it comes to PTM prediction by the training data set. So if you haven't trained on a particular PTM, it is very hard for a model to predict that PTM. And the reason for that is that these models usually don't have an understanding of structure. They just know amino acids and modified amino acids, and there's no relation whether one modified amino acid is similar to the next. So here we have 2 modifications, lysine acetylation and propionylation. I'm sure you can all appreciate that the difference between the modifications is minute. And we have now in INFERIS 5, for the first time, an architecture that can encode these structures and learn from the similarity of these 2 molecules. What does that mean? We do not encode these molecules as graphs, as you would maybe naively do it, but instead we are doing this as molecular strings. We're using selfies here, so this peptide sequence, this peptide with the sequence PEPT is encoded by a string that looks like this, and if you want to, the nitty-gritty details of how we transform this string then for the model, then please go to Lizzie's talk later. Essentially what this means, though, is that we have now, with this model, a whole host of additional PTMs that we can predict. Honorable mentions here is methylation, a whole host of biotin modifications, there's citronylation here, crotonylation, so hopefully some of these PTMs are of biological interest to you, and I hope also that you can appreciate that the spectral angle with which we predict these new PTMs is as high as the previous spectral angles that we were able to achieve with INFERIS on phosphorylation, ubiquitination, and acetylation. All right, now switching gears again, sort of rattling through this quickly, and telling you a little bit about the discovery mode. So far, CHIMERYS processing workflow looked like this. We started out with intensity-based scoring with our predictions. We recalibrated our models. Then we performed a first-pass search and finally error estimation via Mockup. And that is all nice and dandy, but the big problem, I say problem, but the big innovation that is possible here is most identifications that one can achieve with this workflow will be highly specific to a specific raw file. What we do now is we perform a second pass search with a reduced search space that incorporates all identifications across all raw files that we've identified in the first pass, and essentially search them again in a reduced search space and perform another error estimation via Mockup. And what that allows us to do is to substantially increase both data completeness, precision, accuracy, and the number of identifications. And it's a very common way of analyzing data, specifically DIA data. If you as you can appreciate from the scheme, so this is the regular DIA, direct DIA or library-free DIA processing workflow. You start with your samples of interest and your DIA files. You search them against a big FASTA file, that is the true big search space. You, of course, search them also against decoys of the same number. And then you perform your first pass search and error estimation with, in our case, Mockup. Then you remove the decoys and end up with a spectral library, so a very much reduced search space. This search space is much smaller than the original FASTA file, and that spectral library is then used in a second pass with, again, decoys that are added to the spectral library in order to end up at your final identifications and quantifications. That's the workflow when you don't produce a DDA-based spectral library. A DDA-based spectral library would fit in here. You have similar samples as your samples of interest. You produce DDA data, and based on those, you generate the spectral library. And basically, this second pass search is what CHIMERYS does now too. And with that second pass search, what we can do is essentially still control FDR. So as you know, we are very adamant about FDR control. And as you can appreciate from this plot of FDR versus entrapment FDR, even in this two-pass search mode, our entrapment FDR is very well controlled. And the reason for that is that our entrapments and our decoys overlap here in the score distribution. And as a result of this new way of processing data, we can essentially substantially increase data completeness. So here we have CHIMERYS 5 with the new discovery mode, CHIMERYS 5 without the discovery mode, and CHIMERYS 4, the previous version. And you can see that the orange proportion of the bar becomes bigger, from 40 to 49%, but also the bar becomes higher in relation to CHIMERYS 4. And we compare ourselves, we compare ourselves here against 3 different software packages that are commonly used for DIA data analysis. And as you can see, we are basically comparable both in terms of the number of identifications and data completeness. As we see here, again, encouragingly, if we compare CHIMERYS 5 and previous versions to these 3 software packages, the biggest bar in this upset is the overlap. The second biggest bar is the one without software 3. So these are very encouraging results, showcasing that now with CHIMERYS 5, you can really dig deep into your DIA data and get nice data completeness as well as nice identifications. I mentioned precision. Precision is also comparable to the other three softwares that we tested. CHIMERYS 5 is here, the new default here, and 4 here. It's very hard to see, but there's an improvement in terms of precision going from 4 to 5, but the precision was already very comparable to the other 3 software packages to begin with. And if we're looking at accuracy now, this is accuracy in known ratio experiments, so a multi-organism mixture experiment, the LFQ bench data set, as you can see here at the bottom. And what you're looking at is the densities of ratios that we calculate between 2 different samples that have different amounts of human yeast or E. coli. The vertical lines represent the positions where the ratios should be. And as you can see, CHIMERYS 5 with the discovery mode is here. We basically are bang on the different ratios and substantially improve from CHIMERYS 4 as these densities become higher. So we favorably also compare in terms of precision and accuracy. That is basically it from my end. If you want to know more about, on the one hand, our deep learning model for FDR control, our deep learning model for PTM prediction or FDR control, then please visit Lizzie's talk in the morning at 9:50. And if you're interested again in FDR control, you're very welcome to attend my talk in the afternoon. And with that, we are at a little question section, if there are any right now. If not, we can, oh, yeah.
It's great to see that you're adding more modifications to the platform. Do you foresee a possibility of adding kind of our own personal modifications?
So with the new architecture that Lizzie's going to present, in principle, this could be possible. The only obstacle that we need to overcome is that the model, as we have it here, requires the structure of the PTM, because we learn from the structure. And at the moment, there's just no user interface to tell us, well, this is how the PTM looks. It's a small engineering challenge, but in principle, a model, as we have it right now, can be taught to predict arbitrary modifications. All right, if there are no other questions, I think we can move on. And it is my great pleasure to introduce Daniel, who introduced himself earlier. Yeah, he will give you a little bit of an idea what the newest features are on our MSAID platform.
All right, thank you, Martin. So switching gears a little bit from our core product that we developed first, CHIMERYS, as a core component for our platform, to the vehicle itself, which is our end-to-end platform for proteomics. And we sat down in 2023 and sketched and designed our vision of a end-to-end processing platform that works in your browser and would allow you to upload data directly from the instrument, process it, and then leave the results on the web in order to access it if you need it, and not have this moving around of data from your acquisition computer to your processing computer, from the processing computer to your user computer where you do the analysis, and basically get rid of all of that and prep ourselves for a future where data amounts are steadily increasing and the wealth of data that we generate basically exceeds anything we can do on local infrastructure. So we launched this 2024, and it features a web-based interface. You can upload your data directly to a AWS-based S3 bucket, which is then paired with parallel compute instances. What is happening is that you can basically drop your files into the browser, you can upload them, you can tag them, you can filter for them, you can modify them. And you can also start searches in your browser, on your phone, in a laptop if you would like to do so. And we, in the back, perform this using highly parallel compute with a cluster that scales from 0 to hero, meaning that if no jobs are running, we scale the cluster down to 0, and then everything happens on demand instances, meaning that we can crunch away also very large studies within a very limited time period. The results are then deposited in a structured result storage where we can, is that working? No. Where we can access it. You can access it directly in the web using a data browser or visualization dashboards that have some capabilities to do volcano plotting, looking at identifications, or you can programmatically access this data using our API-controlled and scriptable API server, and I'm going to show a couple of use cases of this a little later. We have actually published a paper on the platform and its design principles and architecture in a special issue of the Journal of Proteome Research in this spring. And if you're interested in more of the technical details behind that and how we deal with that processing, please have a look at that paper. But now, moving a little bit into what's new since the 2024 release. I mean, software as a service platforms have the advantage that it can roll out updates relatively frequently, and we've been doing so in the last year. And I want to highlight a couple of key changes that are now available on the platform. Martin already said it, we're launching CHIMERYS 5, and there's a beta version online on the platform right now. We're expecting to enable the timsTOF capabilities within the next couple of days. We worked drastically on improving the scaling in the parallel compute, and I have a couple of examples on that later. We're working on making the data more easily accessible to people, be that using CLI clients for operating systems or R packages for programmatic access. And we have been working a lot on data security and compliance in order to cut red tape when trying to install some program like the MSAID platform within your organization, which might not be as cloud ready as you think. And in the end, we're also introducing changes to our demo tier structure. I have a couple of slides on that. So going through that, as said, there's going to be a new file tab where you can upload and organize your Bruker files just as your thermal raw files. And if you use the CHIMERYS 5 beta, you can process these files just as any of the thermal raw files that have been were able to be processed so far. And the original beta version is online right now, and we're expecting it to mature within the next coming weeks to a final release version. I said we worked a lot on scaling and parallel processing, and we can now demonstrate that it doesn't really take a lot more time to process 1 or 10 files in the cloud. And even if we go to 500 files, and if you look at the wall clock time, you see that if you divide the number of raw files by the total amount of 91 minutes it takes to process these files, we're down to mere seconds for such large studies if you really need that parallelization. We've also demonstrated on that on a little more real-life use case. This is a study from the CRISPR lab where 29 healthy human tissues were deeply fractionated and then measured in order to generate a draft map of the human, like a map of the human proteome. And I remember that back in 2018 when I was loosely involved with this study ran for hours and days on a beefy MaxQuant machine. And I think it took 3 weeks to process this, generate that search result. By now, using that heavy parallel compute in the cloud, we can do this in actually under 4.5 hours, and that includes match between runs and quantification in MS1. Well, I mean, downloading data from Pride, uploading them, and processing them together in a short period of time is all nice and dandy, but what people are worried about is whether these cloud-based services and their upload speeds of their internet can actually keep up with the instruments they have in their basement. And this is a great time to showcase our CLI client here, which is basically a little command line tool that you can use to upload and start searches and also download results of files. So you don't have to really interact with the user interface at all if you don't want to. And so we came up with a little mock study, and as we don't have the plumbing in our office to install an Astral or two, we took data from a study where they looked at brain samples from 72 patients, and that data was acquired on an Astral every 7 minutes, so relatively short gradients in an Astral. So we basically said, okay, let's pretend that this data is generated by 2 Astral instruments, meaning that we're finishing the acquisition of a file type every 3.5 minutes. We watch a folder on our network drive with a CLI client looking for a certain buzzword, and the moment a file appears, we upload it and we start searching it with specific parameters, with specific raw files. And what you can see is that over the 5 hours of data acquisition, we basically crunch away all of these 72 raw files, and in the end, where is the laser pointer? In the end, we're basically 30 minutes after the data acquisition finished, we're also finished with searching all of these files. But then what do you have? You have 72 individual searches, but you definitely want to have match between runs. You want to look across your raw files for protein groups. And we can easily do that because the platform stores intermediate data of your results in a way that we can combine experiments later. And this combination of experiments ends up at the very exact same PSM number, peptide protein group number for runs, no matter if you search them individually and combine them later, or if you search them together in the first place. And this is really neat, because it just takes another 39 minutes to combine these experiments together, and your study is finished. If you would have searched it all together, that would actually be doable in a bit over an hour. I think this really underlines that it's definitely possible to utilize a currently existing infrastructure to just upload and run the data as it comes off the instrument. And it's basically all up to starting to adapt these kind of models. Next, we are also introducing a R package, and a Python package will follow soon. So the idea here is that we have some visualization capacities on the platform. You can look for simple identification numbers. You can perform simple testing, and this visualization capabilities, they will grow over time. However, we also know that most people like to download the data and then look at it using R, using Python, and we're trying to make this a little easier, and instead of having to download some kind of a text format into your user folder and then running your scripts, we're introducing a package that directly interconnects to the data lake. You log in, you list your experiments, you download whatever you want, and then you just start plotting in your favorite environment. And in this case, for example, we're downloading the results of a multi-site plasma study that was done in a couple of laboratories around Munich on different setups. And we can, for example, look as to how different these results actually are, even though they're measured in the same lab on just two different instruments, and work from there. Going away from technical points to a bit more of a high-level topic is we have talked to a lot of people, and we have heard people as it comes to cutting red tape in order to adopting these kind of solutions. And in order to do that, we have built in the last year a state-of-the-art information security management system and have gotten that certified by a renowned certification body. And that means that the development and also the operation of our platform right now has been certified in terms of data security. We also have made great progress in terms of GDPR compliance. We have an external body that is consulting on us with that on us. The platform itself right now is completely GDPR compliant, and we have also added a data processing agreement into our standard end-user licenses agreement, meaning that if you want to give us data that actually contains personalized identifying information, you may now do so, and we have contract work ready for you if that is a topic that your compliance team is concerned about. And last but not least, we also realized that as we're adding features and adding features along, it's important that people keep their account on the platform and can use it from time to time in order to familiarize themselves with the new features, which means that we are switching over our model where you can register a free account and that would be locked out after 30 days, but we're switching these accounts into a limited free mode that will still be able to process a bit of data every month, and the account will be active, and you guys can try it out, can see if the changes to the platform make your life easier, and if there's now something that is worth adopting. So if you're curious, feel free to go to platform.msaid.io if you have already done so. I know a lot of you guys did, and try it out. We also have a booth at 203 the whole week down nose to the main entrance, so talk to us if you have any questions. We can also set up a bit of a bigger demo studies on a case-by-case basis. And with that, I would like to thank all the people that put in the hard work in the back end and the front end to all of this. I'm just the guy who's talking about it. And I'm ready to take a couple of questions. And with that, we will slowly but surely end our product presentation and then afterwards move into our science part. Thank you so much.